Now we're going to review some more topics in vision. These topics are going to focus on neural networks in the visual system and how they might work. It'll start with some very general concepts, and then we'll go to more specific ones that deal with vision. Let's start here with the retina. The retina is that layer of cells. It's on the back of your eye, and the retina contains the receptors, and light energy is going to fall on them, and the receptors to detect the luminance, the brightness of the light, and then there are detectors that detect the frequency of the light. Now, if you look at this graphic, you see that the receptors, they have a convergence pattern here. They converge on that middle layer that you see there, called the inner nuclear layer. And you can see there are cells there. There's also horizontal cells that you see across there. There's a red one there and a green one. You can see, though, that the receptors are converging on a single cell. So a whole bunch of them there are converging onto a single cell, and that information is conveyed by the internuclear layer, which converges on a set of ganglion cells. The ganglion cells become the neurons that make up the optic nerve, and they convey the visual field information back to the brain. But you can tell that there's a process here where you have lots of receptors, and then there's a convergence onto a smaller number in each set of the others. Now I'm going to convey in more general terms what convergence patterns are like and what kind of processing they render for the nervous system. Here's a schematic showing convergence. Now notice you have a neuron at the top, or a set of neurons at the top, and in B and C they're converging on a single neuron at the bottom. Now A is showing you a direct connection between a neuron and the next one. So the axon of the one at the top connects to the cell body of the one at the bottom, and the axon of A fires. In B, you see a small convergence. Obviously, it's just two cells converging on one. And then C, you see a large number of cells converging on one. Now, networks like this, you can imagine the complexities these could have if you had 20 or 30 neurons or 200 neurons or sets of neurons in convergent patterns like this. You can get all kinds of gating and filtering of the signal from the retina, the receptors in the retina. So if you need, suppose the neuron at C only needs two of those to fire in order for it to fire. Well, there you've got a nice filter, a nice gating mechanism that's very reliable. So you need a pretty strong signal or set of them to make the neuron in C fire. Imagine in B, you just need one or the other of those neurons at the top to fire. You can see we could have all kinds of patterns associated with that, or sets of neurons like this, each responding to one or the other input. Well, that's what the retina is like. You have receptors that converge on neurons, and now I'll talk about that in a little more detail. But convergence is this pattern of multiple neurons converging on a single or a set of neurons, causing the subsequent ones to fire given patterns in the receptors. Here you have the relationship of convergence for the neural networks in the retina. So again, on the right there you see patterns of convergence, and on the left there are numbers corresponding to how many actual rods and cones converge on ganglion cells, the nerve cells, that convey information back to the cortex. Okay, you have 120 million rods. Now that's a lot of rods. Imagine them distributed all back there in your retina as individual cells. And you only have 6 million cones. Again, these are all distributed back in your retina. So if you imagine these are distributed, of course, all the cones are in the fovea. There's still cones out in the periphery, a fair number, but the fovea only has cones in it, no rods. So your resolution of vision tends to be defined by how many cones, how dense they are in the fovea. Well, at any rate, you have that's basically the resolution of your vision. When light is focused back on the fovea, all the millions of cones that are back there in the fovea determine how sharp your vision is. Next, you have 120 rods to one ganglion cell. So remember, the ganglion cells are the ones at the top. They're neurons that convey information through the optic nerve back to the brain. So you have 120 rods to one ganglion cell. That's a big convergence for rods. Then finally you have 6 cones for one ganglion cell. Now you can see a big difference there. 6 cones are going to activate 1 neuron. So obviously your nervous system is very attentive to the cone patterns. The information that the cones convey to the ganglion cells are obviously much more important than the rod information. Now rods, however, are more sensitive to light because there's more greater convergence. And there's a spatial summation of the signal, which makes a small level of light stimulating a few rods very influential on making a ganglion cell respond. So all this greater convergence allows them to be much more sensitive. The ganglion cells associated with rods are going to be firing more because light levels, if they go just above a certain level, the rods begin to respond. They stimulate the ganglion cell. One of them is going to stimulate a ganglion cell, and it's going to fire, indicating there's a luminance out there. Cones are not quite as sensitive, but again, cones are conveying frequency information essentially, and their stimulation is going to be regulated by the frequency data that's in the signal. That you want to be more modulated. You don't want to have it to be too sensitive. Otherwise, the frequency information will be sent in an inconsistent fashion, and our perception of color will be built from this, and so it'll be incorrect. Cones are sharper in a sense. When they're stimulated to a certain threshold, a ganglion cell will be stimulated if just 6 cones are stimulated. And so the resolution of the cones is much higher. If you got any of a number of 120 rods being stimulated, the resolution of the light signal can't be that high. It could be, on rod number 5 through 20, or rods number 110 through 120 get stimulated, and they all converge on one ganglion cell. Now that one ganglion cell is going to tell the central nervous system there's some luminance somewhere in that area. But a cone being stimulated, since there's only 6 cones per ganglion cell, you have a much higher resolution of the spatial location of the stimulation, and so the cone cell will be in a specific place. And since there's only 6 of them to one ganglion cell, when the ganglion cell fires, it's going to be roughly indicating a small area of space that those 6 cones reside in, or one of the 6 cones reside in. That gives you a much sharper image. The image now has a higher resolution. It's much clearer. So the rods are more sensitive to light, but they give you a blurrier image. The ganglion cells, the cones are less sensitive to light, but when they're stimulated, they give you a much sharper image. A little later in the course, when we study people that have color blindness, you'll notice that people that have achromatopsia, that is, they don't have any cones, their vision is quite blurry. They need visual aids to see. They can read if they magnify the light and things like that, indicating that, of course, their rod sensitivity is, or their rod sharpness based upon rod stimulation is poor, so they have blurry vision. They fortunately they also though have very sensitive vision. They can see things at very low light levels, like at dusk or even at night, that people with cones don't notice or can't see. So they have blurrier vision, but they have much more sensitive vision. Here's a schematic showing the convergence patterns of rods and cones, supporting the idea that rods have more sensitive light perception, but poorer resolution, whereas cones have less sensitivity, but better resolution. So if you look on the left there, you can see, well, anywhere in that array of 5 receptors at the top will stimulate the one neuron. So light could be anywhere in that space there and stimulate that one neuron. So even though you've got good sensitivity in that network, you have very poor acuity or resolution because the location of the stimulus is not clear. It could be any one of those 5 places. Well, if you look at a rod network, or one where the convergence is much lower, you can see that when light stimulates one receptor, you get one neuron firing. And the location of the neuron firing, the location of the light information, the light frequency data, the light transmission, is clear from this. It's much clearer than in the network on the left. But this network is less sensitive. It doesn't fire as often at low levels of light illumination. But when it does, you get very specific information about the location of the stimulus. Here in this schematic, you can see how convergence helps to form a network that allows the brain to detect the dimensions of a stimulus. the amount of area on the visual field that it encompasses. Notice that when you have a network without convergence, cell B there is just going to fire 4 fires. And the others, they're going to fire consistent with the individual receptors at the top firing. But now if you put B in a network, you can see that there's going to be a different firing rate of B depending upon how large the stimulus is. So if the stimulus only stimulates receptor 4, you get a very low firing rate. If it stimulates 3, 4, and 5, you get a different firing rate. If it stimulates 2 through 6, you get a different firing rate. And finally, if it stimulates all the receptors, you get a higher firing rate. So now the firing rate of neuron B is going to reflect how large an area the luminance is. So you have a large light source that's illuminating a number of receptors in the retina. Cell B will fire consistent with, its firing rate will be consistent with the expanse of the light stimulus. In that way, your brain is going to get information about how large light sources are, and it's going to formulate models, it's going to formulate an idea of forms based on this. And this will allow your brain to perceive how large an expanse a form takes up. And that's important to localize objects in space. The amount of space that is, or the amount of luminance taken up by an object on the retina is a strong clue to everything from the object's size to the object's motion and things like that. Okay, here's another network that uses convergence. Imagine you've got a situation here, you've got 7 receptors, and you've got 3 neurons connected to them. Now, neurons A and C are inhibitory. Okay, if they're stimulated, they inhibit B. They make it less likely that B will fire. So in this situation, you've got these 2 inhibitory parts of the network there. If a light stimulus crosses 3 through 5, you'll get the strongest rate of responding because A and C haven't been stimulated. The receptors attached to them, which normally would inhibit B, haven't been stimulated, and so B will fire a lot. However, once you hit 1 or 2, or you hit just stimulus 4, or you hit 6 and 7, you're going to have an inhibitory influence on B, and its firing rate will be influenced by that. Now imagine if you've got a moderate length light stimulus that passes across this network. It passes back and forth across this network, like you have something moving in your visual fields. You're watching a baseball go through the air. Well, the luminance associated with the baseball is going to pass over this network, and the firing pattern is going to be associated with the inhibitory and excitatory patterns as it passes across. Well, that's a strong clue that something's moving across your visual fields. And this network also gives you a sense of the size of the object. So not only can you detect movement across the network, you can detect its size by the firing patterns of convergence networks that are in the retina. And also other parts of the nervous system, in the central nervous system in the brain, there's also these networks that do the same thing. Okay, now we're going to take neural networks a step further and study how they're going to be used to detect the movement of lines across the visual field. Now, the classic experiment with this, you have a cat in which you've got an electrode monitoring neurons in the occipital area of the cat's brain, where the visual fields are, in the back of the brain, in the occipital lobe. Now, cats have very good vision, and so they have a good map of the occipital lobe. Their retinas are very well developed. Well, here you've got a schematic down below showing different spots, different locations in the visual field, A, B, and C, on the screen, and they correspond to different spots on the retinal image. Now, as the line in front of the cat moves, it's been discovered that neurons respond to that line moving in one direction or the other, and neural networks are undoubtedly the way in which the neurons are configured in order to do this, and we'll show this in a little more detail. Okay, in order to do this kind of discrimination, to show forms in the field and to show how the forms are moving, networks are configured to form receptive fields in which you have excitatory centers usually and inhibitory surrounds, or you can have inhibitory centers and excitatory surrounds in areas of the retina that detect the movement of lines and forms. Now this is beginning to show us some of the early an early general aspect of the central nervous system and how it works. Basically, neurons configure themselves into networks that respond to a dynamic balance of excitatory and inhibitory information. And that's how the nervous system modulates signals. It's how it processes information. It's how it makes your body move and things like that. There's always an excitatory influence and an inhibitory one, and the balance is what produces coordinated motor function or coordinated perception or clear perception. And this is the first example that we're going to see of this. Well, here you've got a visual field, a receptive field, in which you've got an excitatory center and inhibitory surround. And these have been labeled here B and C. And A is the general area of the outside this area in which you've got inhibition and excitation together. Well, this is showing a neuron in the central nervous system. Essentially, if a stimulus is in the area of A, there's no firing or very low-level firing. Neurons never just sit there not firing at all. There's always some excitation of them, but here it's a very, very low rate. Well, as a light stimulus, if it just hits B, there's increased firing. You can see that B there is producing a high level of firing. As the light source goes across C, it turns off this neuron and inhibits the neuron. And so if the light stimulus is only in C, it turns it off completely or brings it down even to a lower firing level than it was at A. But you can imagine a light source being partially across B and C, producing a firing rate consistent with that location. Now, as the object moves across this field, the firing rate of the neuron will convey information about the location of the light. And if there's a line there or a form margin there, the contour of a form, then B and C's firing rate is going to give the nervous system, the brain, very important information about the location of that line. And now we're going to go see how this works with the cat. So now we have a very simple convergence schematic depiction of this. And this is depicting the phenomenon of lateral inhibition, where you've got a center area that's excitatory and a surround area that's negative, it's inhibitory. So that's why it's referred to as lateral inhibition. The outside lateral parts, the sides of it, are inhibitory. The center is excitatory. Now imagine you've got an electrode on the neuron at the bottom. As the light source goes back and forth across this, imagine a line, a dark or white line going back and forth across this network. You see you're going to get a firing rate out of that neuron at the bottom that's going to be consistent with how much of the receptors in the center are excitatory, have been stimulated, versus those that are in the surround. If the line is right over the middle of this network, it's going to get the maximum firing rate. If the line, this is assuming the line is white, so you have a luminance, a line that's light-colored. As you pass away from the center, you'll hit some of the surround area, and that's inhibitory. And so it'll inhibit the neuron from firing. Well, as this line moves around, you're going to have these firing patterns in that neuron indicating the location of that line. Now, this also emphasizes the margin of the light levels. So imagine if you've got a white line on a black background. This firing pattern is going to tell you very compellingly where the edge of the line is, where the margin of the form is. So the nervous system right here at the receptor level is primed for finding margins and contours. And what happens is we tend to use this information to sharpen the image and make the contours really stand out from the backgrounds. That's obviously very important because we're going to be, if you're going to operate on objects, if you're going to reach for them or you're going to be operating on a moving object, you need to know where its margins are. You need to know where its contours are. So if you're going to grab it with your hand, you know where to grab it with your hand to hit the margins, the edges, in order to capture something. Or, if you're swinging through the trees and that line happens to be a branch of the tree you're trying to grab, you want to make sure you get your hand right around the margins of it so that you can swing to the next branch. Well, anyway, the lateral inhibition allows you to perceive margins well. It allows you to perceive where the light and the dark levels change, because you're going to get a different firing pattern when that line goes back and forth, it indicates exactly where that light change has occurred. Here's an image of an animal that's used to study lateral inhibition in vision. This is the limulus, or the horseshoe crab. If you live on the East Coast or near the East Coast of the United States, you've probably seen these before. They're a very, very primitive creature. They have a very, very primitive visual system. And they just have a few neurons in this system, and so it's been a good model to study how things like neural networks work in a real environment. Now, horseshoe crab is an animal that lives on the bottom of the ocean or a bay, usually a tidal bay. If you live near Delaware, New Jersey, you know that the horseshoe crabs come up on full moons, I think in October, to reproduce in the millions. So they'll come up on the shore on the night of a full moon and reproduce and produce billions probably of eggs, and the shorebirds are sustained by them. Back in the old days, they used to harvest up all these horseshoe crabs and actually use them as fertilizer. They've stopped that. Now that doesn't happen, but they were considered kind of sort of a bounty for the farmers to get cheap fertilizer. At any rate, they're very, very primitive. They're from a very early time, primordial time in evolution, and they have a very simple visual system. Okay, well, the visual system consists of a very simple eye. Now, all this animal has to do is basically tell where the sun is, where above is, and where below is. So it needs to swim down somewhere that's darker. It needs to go. If it wants to go to the bottom of the bay, it knows that's away from the sun. It's darker. But the other thing this animal has to do, of course, is detect when there's a full moon, because that's the signal for these animals to come up to the shore and reproduce. And that's the only signal they give each other, or they all correspond to as a group. It enables them to come and reproduce at a certain time in the month. Well, to be able to detect a full moon, they have to be able to detect light levels, and they do this with this simple visual system. Well, their visual system is not really called an eye. It's called the ommatidia, and the ommatidia are composed of a single lens covering a single receptor, similar to an insect. Insects actually have more sophisticated eyes than this horseshoe crab does. But basically you have a single lens covering a single receptor, and that's connected to a network of neurons, which I'll show you in a second. Okay, let's see how the horseshoe crab uses lateral inhibition to see better. Well, up above you have a set of receptors that are in the lateral eye of the crab. And you can see there's a set of receptors. Each one of those has a lens on top of it. You can also see that there's a lateral plexus that connects them. Well, imagine you've got this layout here, and you put an electrode and monitor receptor A. So you see there's an electrode coming from A. Well, if light hits A all by itself, you get a pretty high firing rate. So that neuron fires, the crab gets information that there's light just on that receptor. Now imagine if you also put light on B. So now A and B are being stimulated. Well, the firing rate changes, indicating there's a lot of light present, that there's a broad area of light that's covering both A and B receptors, which is very different than the narrow part of A that was stimulated before. And then finally, if you increase the light level at B, so you have both A and B being stimulated, but then increase light intensity at B, because B is inhibitory, it brings down the firing rate of that electrode at A. Because the lateral plexus there is connecting these receptors, and B happens to be inhibitory in that system. But now you see where you've got a system here that can detect the movement of a light source across this set of receptors. As the light source moves across them, it will stimulate different areas of them, and the firing rate of the cell will change. And so if the firing rate is low in this particular situation, the crab's nervous system gets information that there's a lot of light present in that area. The light is covering that whole set of receptors, and that pushes down the firing rate of receptor A. Now we're going to see how a lateral inhibition produces an optic illusion. In this slide we see the Hermann grid. Now the Hermann grid consists of these black squares and white lines. And if you look at it, you'll notice that in the periphery around the sides of your central vision, you will see ghosted gray squares at the intersections. Now the big question is, why are the ghosted squares at the intersections and not elsewhere on the white lines? Why is it the intersections that they appear? And it's obviously something that's involving the peripheral parts of the retina, because when you look directly at an intersection, the gray square goes away. So whatever's happening at your central part of vision is not happening in the periphery. The peripheral parts, of course, now is rods and not cones. And probably this involves the perception of these gray squares because of lateral inhibition involving the rods. Okay, well, the theory about how this works is that you've got a receptor, A there, A in the schematic part on the right there. You see there's a receptor labeled A, one labeled left and right, L and R, and then one above and below A. Okay, we've got to explain why A is going to be perceiving a lower light level, hence a gray ghosted image. Whereas if you look in the corridor itself, not the intersection, we have receptor B down there, you don't see a grayness. You only see grayness at the intersections. Now the theory goes that these receptors that are around A, when they're stimulated by illumination, by a white area, they inhibit A. So what happens is when left and right and the top and bottom there are in the corridors, that's where there's light, that's where it's being illuminated. However, they're inhibitory of A. And so when they inhibit A, it fires less, presumably, and that lower rate of firing for that receptor gets translated by your brain into less illumination. Hence, it turns gray. Now, in the corridor, you have receptors around B just like at A, but the receptors around B, half of them are in the dark squares. So they're not inhibiting B. B sends information that the line is bright and white. It doesn't get inhibited by L and R there, and so the summation of this inhibition, this lateral inhibition, is that the corridor is white and is illuminated. At any rate, you can see how these patterns of illumination can produce some misperceptions. Now, the thing about optic illusions, of course, is they don't usually exist in the real world. You probably have never noticed that there are gray squares and intersections of grids like this, because this kind of display is pretty rare in the world. It doesn't produce an impairment of your vision because it's such an unusual display, and this is obviously constructed to elicit the optic illusion, and it's not a natural perception. Okay, here's another optic illusion. It's referred to as simultaneous light contrast, and this also involves lateral inhibition. Now, the general principle here is that your receptors and the neural system they're in are forming a dynamic balance of excitation and inhibition so that you perceive margins better, the margin between light and dark better. And your brain will actually manipulate perception in order to enhance this. And here's an example here. If you surround an area with dark or bright backgrounds, you'll change the perceived brightness of the area in the center that's surrounding it. So if we look at these gray squares here in this display, they're actually the same luminance, okay, the same level of grayness. Yet as I look at this, I see that the left one appears darker than the right one. Now this is going to be a misperception of lightness, of luminance based on lateral inhibition. Because see, the area around the bright area will be stimulating. It stimulates the receptors, stimulates that system, which may be inhibitory. And the dynamic balance produces a grayness level for the middle part of a certain value. And here it's darker. But around the gray on the right, it's dark. It's black around that outside of that gray square. Well, if there's not a luminance, if it's dark, it's not going to produce any stimulation, and so the dynamic balance allows the brain to perceive the middle part of this display as lighter, as if the luminance is higher, because the receptors in that area are not being inhibited by the area around it. Now you can see how this works. This dynamic balance of inhibition and excitation produces a contrast enhancement that enhances the margins between the light and dark areas. See, the strength of this kind of illusion, even though it's slightly inaccurate, is it enhances the margins of that middle square. Now, if you can imagine that middle square is an object that you have to grab or touch or you have to operate on, knowing where its margins are, knowing where its contours are, is obviously going to be very helpful. Well, at any rate, lightness contrast changes, this dynamic balance between light and dark, and margins of light and dark, give us that ability to form perception of contours and lines better, and that enables us to interact with objects in the environment better. Here's a schematic depicting what I just described in the previous slide. Now you see you've got a sort of a schematic of receptors underneath these displays. Where there's luminance, on the left there you see, where there's luminance, there's a higher degree of inhibition. And that higher degree of inhibition is formulated by the brain. The inhibition causes a firing rate drop in the receptors in the middle. And your brain interprets that as if they're not being stimulated, and so it makes that area darker. Well, on the right side, where you've got darkness around the center, there isn't as much inhibition. And so the brain interprets that as, since the firing rate hasn't changed, it interprets that as if the luminance is higher. And so we have this slight misperception. But again, the advantage of this misperception is the margin between the gray and the gray area and its background is enhanced. We have a much clearer, it's almost like it's a filter that's sharpening this making the contrast higher so that we can see the margin of the gray and the background better. And again, that perceiving of margins is very helpful for interacting with the environment. Here are some further illusions that are produced by lateral inhibition and enhancement of contrast. Here you have the binary cross and White's illusion. Now notice that when you look at the cross down below, you have some kind of higher order processing of these contrasts because this can't be accounted for just by lateral inhibition, but it does produce a simultaneous contrast. Notice that if you look at A and B, the gray values there, A looks darker than B. Now the surrounding areas for both A and B are equally dark. So the darkness ought to produce low inhibition, hence they should both be equal in terms of their luminance, but they're not. And the reason proposed is because you have your brain has such a compelling system for enhancing contrast, enhancing forms, it makes B part of the cross. It makes A an object either separate from the cross or part of the background of the cross. Well, when it does that, see, it wants to make B part of the cross, and when it does that, the lateral inhibition in the periphery produces a lighter luminance for B. If A is not part of the cross, then it's not going to be influenced by the darkness around the cross. And so your brain is going to reinforce that it's not part of the cross by making its luminance based probably on the background. In that case, it makes it darker. At any rate, it's still quite a mystery how this works. It's obviously some way the brain is trying to reinforce forms and shapes. That cross is a very compelling shape and it wants to maintain its integrity. And so it makes B part of the cross, since it's B's margin configures to a cross, so that you can perceive the cross better against the object of A or the background that A is part of. And that way, if you need to operate on the cross, you clearly know where it is. Now, in the White's illusion, you see again a similar problem in that once you make part of the gray values part of an object, it now is influenced by that object and not by just lateral inhibition. So you can see where I perceive A clearly as part of the background. B, though, the gray area of B, I perceive as part of the bands across. It's as if B is made up in a rough way of the crossing bands, and I give B a luminance based upon that, and give A an illuminance based upon it being surrounded by the background of whiteness. Whereas the B area, even though all those B blocks are surrounded mostly by black, they're made to be lighter because they're part of it, part of the black part, and not part of the background. Well, all these things that you see here, these illusions you see here, are mediated by the cortical systems that process vision. And again, the overriding consideration is to create forms in the environment, to create contours, create objects in the environment so that you can operate on those objects. And if you keep them as they are in the world, the contrast isn't going to be sufficient for you to clearly perceive objects the way you can if this process is in place that enhances margins, enhances what an object is versus its background. This is going to be a theme through a lot of parts of the course that deal with vision in particular. Vision has a very compelling model in which it wants to keep, you want to keep objects in the foreground and objects in the background and make good estimates of distances between them and things like that so that you can actually form a spatial model of the world that's accurate. It's actually enhanced because you can perceive contours so well. Here are some other examples of how cortical processing influences the perception of an object. These are ambiguous figures in which the contours are incomplete. Now what you should do is look at them, look at these figures, and the next slide will be the correct figures, or the completed figures. And perceiving the completed figures, when you go back to these, should enhance perception of these. And so what you're going to do, what your brain is going to do, once it sees the completed figure, it's then going to have a complete model of what this thing should look like, and it will impose that model on the stimulus to the point where you can't see this as anything else. If you look at the figure on the left, it looks quite ambiguous. It looks like nothing, really. It looks like there's maybe some clouds or something like that. The figure on the right looks like some kind of an object. Maybe it's a telescope or something. It looks like it is. It's got a very firm outline to it. It looks like the bottom part on the right might be a shadow, maybe, of what the object is. At any rate, look at these, and we'll then go back to them to see seeing the completed form, how that might help you perceive these now. So go to the next slide, then come back to these. All right, in this slide, you see the completed images. So you see that on the image on the left there, it's a cow staring back at you. And here you see the background's been filled in black, and the cow is pretty clear. You also see on the image on the right a sort of kind of a nonsense image there. I don't think that's a real object, but you can see that it actually is a shape, a form there with a shadow. Now go back to the previous slide and look at those pictures and see if these images don't prime you or compel you to see the cow and this object when you look at the ambiguous stimulus again.